Science 101 introduction, and then I'll talk to you a little bit about some of the work I've been doing over these years. So let's get right into it. And as mentioned, if you have any questions, feel free to write them in the chat, and then at the end, I'll be delighted to tackle those. So as part of my work, I spend a fair amount of time studying amphibians during winter. And the reason why is because we are in Ohio and low temperatures usually occur during winter. I guess this morning's frost excluded. But I would like you to now get into the frame of mind of a frog, of an amphibian, and think about what are some of the challenges that you may encounter during the winter season. So, of course, spoiler alert, low temperatures are going to be the big focus of this talk, but there are other things that amphibians have to struggle with during winter. So some of the things that you may consider, in addition to low temperatures, are just a generalized lack of food. Frogs are carnivorous. They feed mainly on invertebrates, so things such as insects or spiders or even earthworms. And so during winter, in general, these types of prey are not really available. So this is one of the things that as an amphibian, frogs have to also handle during this particular season. Not only do they experience a generalized lack of food, but there is also a general lack of water. And so you may be thrown off by the fact that I just put here a beautiful picture of a snowy mountain, and you may say that's water everywhere, and you're right. But the problem is water that is frozen as ice or snow is in general unavailable to most animals. So it's not like they can basically melt that to obtain liquid water. And so in general, winter is also fairly challenging because of this general lack of water being available. And so that will become relevant also when we talk about freezing and some of the stresses associated with that particular physical state. But then, of course, last but not least, low temperature is going to be the major issue that these animals are going to experience during winter. And due to some physiological features of frogs that we will talk about next, freezing is something that is a very, possible reality for these animals. So let's talk about why that is actually something that they have to deal with. So frogs are part of this group that we generally call cold-blooded organisms. So it's a little bit of a misnomer because it's not that their blood is cold. We usually call these organisms ectotherms. So I'm assuming everyone in this Zoom call right now is a mammal and a human. And so your body temperature is around 98.6. That heat, those 98.6 degrees Fahrenheit, are generated by you. There's a series, basically the chemical reactions that your cells are constantly doing help to generate that heat. Things like movement help to generate that heat. And then also, when you're digesting food, you generate some heat. So there's a series of physiological processes that help to generate a massive amount of heat that keep you warm. However, the group that we call cold-blooded organisms, we call them ectotherms, they do not have this ability to generate massive amounts of body heat. And so instead, they rely on an external heat source, hence that ecto, which means outside. Part of their name, ectotherms. So their heat source is usually the sun. And so what this really means in practical purposes is that an ectotherm, a cold-blooded organism, if it's in a room that is at 75 degrees Fahrenheit, the body temperature of that animal will be at around 75 degrees Fahrenheit. However, if that same room is instead at 32 degrees Fahrenheit, the body temperature of that animal is going to be at around 32 degrees Fahrenheit, because again, they're dependent on external heat for their own heat. So if you're in this particular situation, if you're an ectotherm, evolutionarily speaking, there's kind of two approaches to surviving winter. So there's the approach of avoiding low temperatures altogether, in which case there's a couple of options. And when I mean options, these are not real options. These are just evolutionary solutions. These organisms can hibernate, so they can go to protected hibernacula, so protected locations where they will spend winter, and in general avoid these low temperatures, so they won't ever be exposed to temperatures that could potentially cause them to freeze. I included some pictures down here to kind of illustrate these examples. So we have here a garter snake exiting its den, so its hibernation location. And so garter snakes usually do communal hibernation. So you have usually a few hibernating together. They go into caves or crevices. Many of my neighbors are super lucky and have garter snakes hibernating in their basements. So by doing this, they avoid exposure to those low temperatures that could potentially cause their body fluids to freeze. Another possibility would be migration. And so we do have some species of ectotherms that avoid winter altogether by migrating to, warmer locations. An example, monarch butterflies. Migration is not usually something that is available, evolutionarily speaking, to amphibians because they can't really move that fairly large distances, but it is a possibility for some ectotherms. Now, on the other side of options, we have different types of ectotherms that have evolved to actually tolerate low temperatures. And when I talk about low temperatures in the context of this talk today, I mean temperatures that would cause the organism's body fluids to freeze. So we are definitely talking about temperatures below the freezing point of water. So we're talking less than 32 Fahrenheit and even lower than that. And so if these organisms are going to be exposed to these sub-zero, 0 Celsius, sub-zero temperatures, so these freezing temperatures, there's a couple of possibilities. And again, evolutionarily speaking, these are paths that are not mutually exclusive, but in many cases, organisms tend to favor one versus the other. So we have some organisms that have evolved to tolerate this cold, but avoid freezing altogether. And painted-turtle hatchlings are an example of those. So painted-turtle hatchlings will spend their first winter actually still in the nest, where they are exposed to temperatures that could cause them to freeze. However, they don't. They are able to remain unfrozen at these very low temperatures. So they're in this state called supercooled. This is a risky business, though, because organisms that are supercooled cannot really be disturbed. And if they encounter ice, they can freeze instantly. And if they do that, they die. So it's a very, there's a trade-off for sure, but it's a strategy adopted by some species and it works for them. And then on the other side of the coin, we have organisms that are going to face low temperatures. And when they encounter these temperatures, they will survive freezing of their body fluids. And so the wood frog is one of the example animals that I will talk about today. So it's an animal that during winter is in locations that have temperatures that drop below the freezing point of the body fluids of this organism. And so this means that if the conditions are right, this animal can actually freeze. Its body fluids can lead to the formation of ice. So why is whole body freezing a problem? I suspect that you have this preconceived notion that being frozen is possibly problematic. But I would like you to think about why. Why is it problematic? And to help you get into that frame of mind, again, I would like you to think from an amphibian perspective. First of all, what does it even look like for a frog to be frozen? It doesn't look at all like this picture that I have here. It is not a frog, kind of a frog. It's not really an actual frog inside an ice cube. But instead, it looks something like this clip that I'm going to show you. Ooh, I have to do something. Give me a second. I have to share this, otherwise we will have problems later for sound. Give me just a second. Let me share this again, but with sound. There we go. Okay. So going back to what I was saying, what you're going to see now is a clip. So it's a clip of a documentary that is filming a frozen frog. Because of the connection speed of everyone, you may see this kind of like pause and continue, but I would like to emphasize that these are not still images. This is, like a continuous shot of a frog frozen and from different angles. So if nothing is happening, if you don't see the frog moving, it's because the frog literally wasn't moving, okay? And I would like to start with this beautiful image, which is of an eye of a frog. So what you're seeing here is kind of like the opaque eye, because the inside is frozen, and you see this beautiful network of capillary hairs capillaries here, and the red is blood. So when I start playing, what you'll see is that the blood is not going anywhere because it's frozen. Okay. So this is what a frozen frog actually looks like. So these animals usually kind of crouch down. There's ice everywhere. You see this, for example, we see here the front paw limb with ice on the digits. You see the skin is fairly dark, and if you handle a frozen frog, you can actually feel ice slabs on the back, on the ventral side, so on the belly side. And the arms are all frozen in position. So it's not like you can articulate it like, you know, a Barbie doll or something like that. It's kind of frozen in position. Okay. So nothing is happening. There's no circulation, there's no ventilation, there's literally no movement. And so considering this, and given that we're talking about an animal that is very cold with a lot of ice inside of its body, what are some problems that, or what are some things that you would expect would be happening in a live animal that are probably not happening in an animal in this particular condition? So when we talk about animals that are frozen, in general, we talk about three major types of problems that are happening, or challenges that are associated with being frozen. And I have those listed here. So the first problem, we refer to it as mechanical stress. It has to do with physical problems that are associated with being frozen, because we're talking about a large amount of the body water of the animal turning into ice, solid ice. As I mentioned, those ice slabs, the limbs are stiff. And so this is quite problematic. If you think about ice that forms, for example, in the blood vessels, these animals are at risk of experiencing multiple instances of internal bleeding because ice crystals can cause ruptures of the capillaries, for example. The ice is forming in, almost like needles within the muscles. And so again, we can have damage to the various tissues and organs. So the first problem with being frozen is you are frozen. There's ice everywhere, except for inside of the cells. That's the one type of ice that most animals can actually not survive. So we don't see that. But you have ice pretty much everywhere else. And so that can be quite problematic. The other problem that we have in frozen animals is this problem of osmotic stress. So where does the water come from to form all that ice? Well, water comes from the blood. There's water in between the cells, but then also water also comes from inside of the cells. And so all of that water is physically being kind of sucked out. It's an osmotic process. And so water is being drawn out of the tissues, drawn out of the cells. And so when you pull water out of the cells, you cause those cells to shrink. And there's only so much shrinkage that any kind of cell can tolerate. And so that is also problematic for these animals. And not only that, but then everything that is not water is kind of left behind. So there's like this slushy of ions. So things like salts and sugars and other compounds that are found in these animals that are now in basically the wrong concentrations, the wrong proportions. Everything is much more concentrated because you've basically trapped all that water as ice. So this is all under this umbrella of osmotic stress. Osmotic has to do with osmosis, with water movement. So problems because you're pulling water from the cells and then you're leaving all of these compounds behind, which are now basically in the wrong concentrations. And that can be also pretty damaging for any organism. And then last but not least, metabolic stress. So this has to do with metabolism. It has to do with the normal physiological activity of cells and organs. As I mentioned, nothing is moving. There is no ventilation. This means that there's no breathing. There's no circulation, so the blood is not flowing. And so things that this animal normally would have access to, such as oxygen, such as nutrients, are really not available because the delivery mechanisms are not there. There's no air coming in and out. There's no blood flowing to the lungs and then delivering oxygen to the cells. And so this means that this organism has to basically rely on less than ideal ways of producing energy. Because yes, animal looks like it's dead and it looks like nothing is happening, but in reality, the cells are still undergoing various chemical processes and energy is still needed. And so there's still a need to produce energy, even in these extreme circumstances. And so what this really means is the animals have to produce energy without oxygen. And that's called anaerobic respiration, or, it's a kind of a form of fermentation. But what it really means is they can produce then compounds from that that can be toxic if accumulated in large amounts. And so there's also that to deal with. So it's bad enough that there's all this ice, you also are trapping this water, and you're creating all of these stressful chemicals that these animals have to deal with, which makes this even a more extraordinary physiological feat. So freeze tolerance, I like to think about it as a combination of 3 superpowers. And so these are kind of generalizations, but usually organisms that are freeze tolerant have to be capable of dealing with 3 different problems. So the first one is low temperature. So they have to be capable of tolerating low temperatures. The second one is dehydration. And if you think about it, I kind of already explained why. Because when these animals are frozen, they lose a lot of their water because the water is trapped as ice. And that water is not available when it's ice. It's like it's not there. So they have to be able to survive in situations with low water inside of their bodies. So they have to be effectively tolerant of dehydration. And then last but not least, they also have to be tolerant of anoxic conditions. So conditions with basically no oxygen or very little to no oxygen. Okay? And so these 3 capabilities come together under this kind of ability of tolerating freezing. So we are in Ohio, and due to some weird evolutionary circumstance, we happen to be in a great state to study freeze-tolerant frogs because we basically have most of the freeze-tolerant species in North America. So freeze-tolerant frogs are the wood frog. We have the spring peeper as another freeze-tolerant frog, the chorus frog, and then tree frogs, specifically the gray tree frog and Cope's gray tree frog. Everything else is pretty much freeze intolerant, so they really cannot tolerate any relevant, intense exposure to freezing. And this includes toads, aquatic frogs, and then salamanders. And so I'd like you to look at these 2 lists, these 2 columns, and think about either, what do the intolerant amphibians have in common, or what do the tolerant amphibians have in common? And maybe if you're like, person that likes herps, in general, you have an idea. So the difference here is, what are these 2 groups doing during winter? And if you think about toads are usually pretty good diggers. And so for winter, what they usually do is they bury themselves deep down in the soil. So usually they can go below the frost line. And so they don't really deal with low temperatures that would cause them to freeze for the most part. If you think about aquatic frogs, they usually spend the winter in the bottom of ponds. So they usually don't deal with freezing either because, in the bottom of a fairly deep pond, the water is never really going to freeze. So they don't have to worry about that either. And then things like salamanders usually will spend, the winter in crevices or even caves and things like that. And so usually they are not going to be exposed to those extremely low temperatures that would trigger them to freeze. So in general, the difference between these 2 groups is some have evolved to spend winter in these locations where, freezing is not really going to be something that they will have to encounter. But then we have these freeze-tolerant animals that have evolved to hibernate in locations that do expose them to these low temperatures. So freeze-tolerant frogs overwinter terrestrially. So there aren't honestly a lot of studies that have gone out and checked hibernation spots of all of these frogs, but there are some. And so the commonality seems to be that freeze-tolerant amphibians overwinter terrestrially, usually in superficial, depressions in the forest floor. So they're not usually particularly good diggers. They're not usually going to be overwintering in the bottom of a pond or even in a cave. So they're just going to kind of lay in the bottom of the forest. Maybe there's some leaf litter on top of them, but they're not going to be very well insulated or buffered. And so this seems to be the circumstance in which they will encounter these low temperatures. Another thing that some of these frogs have in common is that they're early spring breeders. So some of these frogs, particularly wood frogs, spring peepers, they're the first frogs to come out, in late winter, early spring. And so the fact that they're at the top of, top of the forest floor allows them to detect when temperatures are starting to change, when we have those nice early warm rains. And so they can detect fairly quickly, that spring is basically coming, and this allows them to, come out of hibernation, and then not migrate, but basically move to the ponds where they usually undergo mating, which is the first thing they're going to do when they come out of hibernation. Now, what are we really talking about in terms of freeze tolerance? Some numbers, like how extraordinary of a capability are we talking about? Well, we are talking about freezing of up to 2/3 of their body fluids, so 66% approximately, and again, this varies by species. We're talking about the lowest temperature recorded around -18 degrees Celsius, so that's -0.4 degrees Fahrenheit. This is approximately the temperature of a household freezer, approximately. I want to say you cannot, however, take a wood frog and put it in your freezer. It definitely will not survive that. We'll talk about that in a little bit. And then in terms of duration, we're talking about, depending on the population, we're talking about some populations being capable of surviving continuously frozen for as long as 8 months at a time. So all of those stresses that I mentioned for 8 months, 8 months with no oxygen, no circulation, no nothing, just ice formed, etc. So think about that. So the question is, how is this possible? How do some frogs survive freezing? What are they doing? Okay. And there's obviously, not obviously, but there's a multitude of adaptations that allow frogs to survive freezing. And today we're going to focus on one of them, mainly one of them, which are going to be these chemical compounds called cryoprotectants. Okay? And to introduce that, I have here a short clip to show you. So let me double check. Alex, can you just let me know if you can hear the sound?
Of all the
we can hear that, yes.
This perhaps is the most extraordinary. It's called the wood frog, and it lives in America north of the Arctic Circle, and it survives some of the coldest temperatures on Earth. It does so by becoming frozen solid.
Many animals can adapt to the harsh winter weather, but the wood frog has a unique method. As the winter frost starts to bite, ice begins to form on the frog's skin. The liver goes into overdrive, producing glucose, which is pumped around the body by the heart. This glucose acts like an antifreeze within the cells, preventing them from freezing. Instead, ice forms around them. The blood, however, is frozen, and all the organs are encased in ice. And the heart stops. But then, months later, spring at last returns. And the ice around the wood frog begins to melt.
The wood frog's ability to survive is truly extraordinary.
Okay, so that was a clip that was actually an Ohioan frog, in case anyone is curious. And so there's a couple of things that were shown in the clip, but the key thing that I would like to focus on is this concept of cryoprotectants. So these are compounds, chemical compounds that are naturally produced by these amphibians that help to minimize some of the damages of freezing. So some of those stresses that I mentioned before, they are going to be minimized by the presence of these compounds. And so again, what do? So they help to reduce the amount of ice formed. A lot of times whenever the media or, like a production company does a documentary or something like that includes freeze tolerant, they like to use the term antifreeze. It's a little bit misleading because there's still ice formed in the animal. What's happening is you have less ice formed than you would otherwise if you didn't have the cryoprotectant. So in general, these compounds help to reduce the amount of ice formed at any given time. They help to limit the water loss. So they help to limit the water that gets pulled out of the cells. So you have less shrinking of the cells. And then they help to stabilize enzymes. Enzymes are basically molecules that help chemical reactions take place in the cells. So they help to stabilize those molecules and also the membranes of cells. So basically the external boundary of the cell is basically stabilized by these cryoprotectants. And so you must be thinking, what kind of compounds are these? So glucose, it's basically the blood sugar found in vertebrates. You have it, we all have it. Glycerol is another example of a cryoprotectant found in amphibians. Urea, these are all compounds that literally all of us have in our bodies right now. The difference is we are talking about completely different levels of these and completely different capacity of production of these, okay? And so what we're going to talk about next is basically what do we know about the production of these cryoprotectants and how does this help us understand the freeze tolerance of a couple of different amphibians. So in general, I am interested in cryoprotective responses and basically how do they respond to freezing? So how are they triggered by freezing? And also other environmental stresses, because as I mentioned, freezing is being able to survive freezing means that you are able to survive many different problems at the same time, and sometimes not even at the same time. And so that's basically some of the questions that I'm trying to address. So I've studied mainly two different species of freeze-tolerant frogs. One of them is the wood frog. And by far, hands down, the wood frog is the rock star of freeze-tolerant amphibians. It has been studied for decades, and it's right now, in terms of frogs, the most freeze-tolerant species that we are aware of. The Ohioan population is capable of surviving, approximately -5 degrees Celsius, that's 23 degrees Fahrenheit, for up to 2 weeks. And in terms of their cryoprotective strategy, they can do two things, or they do two things. So they accumulate urea as a cryoprotectant, and they do this well in advance of any freezing episode. So they start accumulating urea during the fall, as temperatures start getting down and the weather, humidity starts also going down. So there's a couple of changes that trigger urea accumulation in these animals. And so urea is something that you would produce if you're using, for example, protein to produce energy. So urea is a product of protein breakdown. And so the difference is you normally excrete a lot of it in urine. And what these animals do is instead they hold on to it and use it as a cryoprotectant. And then the other interesting ability that these frogs have is they also produce glucose. And as shown in the documentary and narrated by Sir David Attenborough, is that they only do it when they need it. So they produce glucose only when freezing begins, which is also quite fascinating that they're able to just turn this switch on and then suddenly they can mobilize all of this cryoprotectant to protect their tissues from the impending ice that is forming literally everywhere. Now, when you look at the wood frog distribution, you can see that it's widely distributed in the United States. And when we're talking about latitude northwise, you can see that it extends even beyond the Arctic Circle. And so for about 10 years ago, I was able to be part of this project where we were looking at how, first, well, there was two questions. I'm kind of getting ahead of myself. So there were these previous reports that wood frogs from Alaska were more freeze tolerant than wood frogs from Ohio. And when you think about it, it would make sense. If you see here in this distribution map, we have some temperatures and also snowfall. And so what you see is that the low temperatures in Alaska are very low, like -28 degrees Celsius. That's fairly cold compared to Ohio, which, -5, which I told you was around 23 degrees Fahrenheit. So we're talking about a massive difference in terms of low temperatures. So you would think it would make sense that the frogs from Alaska would be more freeze tolerant, but in reality, we didn't really know. And so we kind of undertook this project at Miami. This was during my PhD, where we were investigating, one, were Alaskan frogs really that much more freeze tolerant than Ohioan frogs? And then if so, what contributed to this extreme freeze tolerance in these Alaskan frogs? So for perspective, this is what wood frog habitat looks like for those of you who are not familiar with these particular creatures. So we usually find them during their mating season. So in the mating season, where you find wood frogs in Ohio is in these, temporary ponds, not particularly deep, in the middle of the woods, and in general in regions that are away from any urbanized areas. So either that or flooded areas like plains, and again, away from urban areas. And so it's not a particularly abundant frog in Ohio per se. There's, where you find them, there's plenty, but there's only specific places where you can find them, at least based on my experience. In Alaska, it's a completely different story. Basically, during mating season, it seems like Alaskan wood frogs are everywhere. This is just a couple of pictures. So we're talking, flooded areas in Alaska, by the river or by lakes, and you can find them, close to cities. You can find them pretty much in deserted areas. So they seem to be very, very abundant in the state. So when they're mating, you can hear their calls pretty easily as you are driving and so on. Now, what are some things that you may see when you go collecting frogs? So I guess I should say something that I haven't mentioned. So I'm a physiologist, as mentioned in the beginning. And so basically speaking, I study how the body works. And so when you're trying to understand how the body responds during freezing, you basically have two options. Either you go out in the woods in the middle of winter and hope to find a frog frozen at a specific point in time, which is highly unlikely that you could ever do, or you can collect these animals and then in the laboratory or under control conditions simulate a freezing episode, and then you can see how the physiology of the organism is responding. And so I do the latter. So I simulate freezing episodes in the laboratory, and then I study how the various organs and how the different compounds in the blood react to these stresses. So this involves collecting frogs from the wild. And so you may be faced with scenes like this. For example, we get to see the animals mating in the wild, which is, from a biological perspective, really interesting behavior. For example, here we have a beautiful red tinted female being embraced by a male. This here seems like a terrible picture, and it is, but it highlights an important point. Each of these white dots is a pair of wood frog eyes. So you can see how abundant they are in these, ponds while they're mating. And it's not only wood frogs that are out when we go out collecting. We see different types of organisms, so different types of salamanders, spring peepers. And so for perspective, when is this happening? So in Ohio, this is happening usually mid-February. So this is not a pleasant experience by all means. It is very cold. It's usually like 50 Fahrenheit. Most of the times it's raining and it's the middle of the night. But still, biologically speaking, it's really interesting and fascinating. And I can't bring you with me, of course, but I have a small clip of what it sounds like and what it looks like when we go out collecting. So I do have a warning. Please lower your sound right now. So phone, tablet, computer, lower your sound. There are two species of frogs calling in this video. You only see the wood frog in it, but you're going to hear two. And one of them are spring peepers, and they are high pitched. So it's going to be painful if you have the volume high. And so the spring peepers sound like beep, 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 like really high pitched. And then the wood frogs sound basically like ducks calling. So look for the duck call, okay? And so this is what it sounds like when you go frog collecting. Okay, I think that's enough. I hope everyone was able to deal with that. Okay, so you hear the high the high-pitched peeper call. It's like really, you know, persistent. And then you hear that quack, quack, quack of the wood frog. And so we're talking about hundreds of frogs, you know, in fairly small areas, and all of them mating. And so this is when we can find our animals to do our research. So as a reminder, what was our question? What am I talking about? So we were investigating basically whether wood frogs from Alaska were more freeze tolerant than wood frogs from Ohio. And if they were, how they did this. So in fact, we did find that Alaskan wood frogs were more freeze tolerant. So that those temperatures that I gave you in the beginning, the negative 0.4 degrees Fahrenheit, that's as low as we found Alaskan frogs were able to survive. And that was not the limit, that was just the lowest temperature we did. And so then it became, you know, imperative that we dug deeper to understand how they did this. And so again, how do we do this? So there's multiple ways to simulate freezing episodes. So as I mentioned in the beginning, you can't just take a frog, put it in your freezer and expect it to survive. It doesn't work like that. So these animals have evolved to tolerate freezing, but freezing as they experience in their environment, which is the temperatures cool relatively slowly, which basically allows the organism to respond to the change in temperature. If you do it too quickly, that's lethal and the animals will not survive it. So here we have, for example, this is called a circulating bath. And so we put the animals in these containers, which are basically these small tubes, and then we can circulate cold alcohol in the bath. And so that allows us to have a fine control over the temperature at which the animal is at, and we can pretty much recreate what would be happening in nature. And so this is just one of the various ways in which you could simulate a freezing episode in the laboratory. So once we do this, in the case of the Ohioan and Alaskan frogs, what we were interested again in comparing is what are the two populations doing that is different. So we subjected them to freezing episodes, both Alaskan and Ohioan frogs, and then we took various physiological measurements. We measured blood levels of cryoprotectants, we measured activity of enzymes in the organs and what the different organs were doing during freezing to try to determine what was allowing Alaskan frogs to be so good at freezing at those low temperatures. So I have here just a little bit of the data that we collected during the years to illustrate some points. So we'll start with the graph on the left. So we have Alaskan frogs represented here and Ohioan frogs represented here. And this is a graph of liver glycogen. So again, when we looked at the little clip of the freezing frog, it was mentioned that the liver goes into hyperdrive. So why do we care about the liver? So it turns out that in frogs, in freeze-tolerant frogs, the liver is extremely important as a source of cryoprotectants. The liver is actually full of stored cryoprotectants. Glucose is not stored as glucose per se. It's stored as something called glycogen, which is basically just a long chain of glucose molecules. And so your liver is full of glycogen and your muscles too. But these frogs' livers are very, very full of glycogen. And so what we found was that Alaskan frogs had in fact much higher glycogen levels than the Ohioan frogs. And not only that, but their livers were much bigger in proportion to their body size than the livers of Ohioan frogs. So it seemed like in terms of just organ size and strategy for storing nutrients, Alaskan frogs were just better prepared. And if you remember, I told you that one of the challenges of winter was the lack of food, right? And so if you think about it, these animals are not feeding during winter. So they cannot get glucose from crickets or earthworms or anything like that. They cannot get any kind of cryoprotectant from the things that they're eating during winter because they're not eating anything during winter. So they have to have all of these stores available to them to create cryoprotectants. And these stores are usually found in the liver. So yes, one of the things we found was that in fact, Alaskan frogs had just in general better stores of cryoprotectants. It then became interesting to determine, for example, okay, we see these whole frogs are different at the whole organism level. So Alaskan frogs are more freeze tolerant than Ohioan frogs, and they store more cryoprotectant. Are they different at a cellular level? If you take a liver cell from an Alaskan frog, and if you take a liver cell from an Ohioan frog, are their freeze tolerances different? Are these cells better than the Ohioan ones? And so we also tested for that. And that's what you see in the graph on the right. So we have again here Alaskan populations and Ohioan populations. And what we did was we isolated liver cells and we just kept them cold at 0, which would be the white bars. We then froze these cells at -4, which is mild. And then we froze those cells, different batches of cells at -8. So we basically gave them different temperature stresses. And we asked the question, are Alaskan frog cells better than Ohioan frog cells And the answer is no. They basically did the same. So even though Alaskan frogs had more cryoprotectant and the ability to produce more cryoprotectant, at a cellular level, they are still very similar, which was an interesting finding for our study. Now, just for perspective, this was a study that involved several students and multiple years, and work of this study is still, coming out. And so some of the things that I'm going to list here are years of work. But some of the major findings that came from that project was we did find an enhanced cryoprotectant system in these frogs. And the most intriguing finding wasn't even related to glucose as a cryoprotectant. It turns out that Alaskan frogs rely heavily on urea as a cryoprotectant, and they accumulated to levels that had never been seen before in this species and in most frog species. And so that was pretty new and had some serious implications in terms of the physiology of these organisms. We didn't find any cellular differences in freeze tolerance in these populations. And then we also did a series of work where we looked at how different genes responded to cold and freezing in general. And we did find that the populations were turning on different genes at specific times. And so right now, one of the things we're still working on includes what is the liver doing to prepare for winter. So which genes are being turned on or off to prepare for the freezing events and other stressful things that these frogs have to deal with during this period. Now, the other species that I work with is Cope's gray tree frog, which you see here, a lovely specimen. And so I worked basically on two things. So one of them is we just finished that work where we were looking at genes and how they respond to low temperature, so to freezing in these animals. And so just a little introduction on Cope's gray tree frog. So we find this frog in the eastern United States. Its freeze tolerance is milder than the wood frog. So they can survive freezing to around 27 Fahrenheit for about 2 weeks. Curiously enough, they have also a dual strategy in the sense that they produce some cryoprotectants much in advance of any freezing episode. So in the fall, they accumulate glycerol, which is a cryoprotectant we hadn't talked about before, and then also urea. And then they also have that capability of producing cryoprotectants when freezing begins. And that's true for both glycerol and glucose. So the other thing I want to mention is the wood frog in Ohio. They usually mate for about a week. So when you want to work with Ohio wood frogs, you have to pay close attention to the weather starting in late January, because you kind of need to catch that 1 week window in which they come out and they're easy to find, because after that it's in general very difficult to find wood frogs. Tree frogs are much more civilized, so they breed for multiple months, and it's when it's warm. So we usually go out collecting tree frogs in June at night, so it's nice and warm. There's fireflies out. It's beautiful. So again, if we have any herp enthusiasts, I strongly recommend that you go out in late June after a nice summer rain, and you'll get to see fireflies, and then you'll hear all of these frogs, and one of them are going to be these tree frogs. And surprisingly enough, they are not camera shy at all. And so again, I have just a little video of what it's like to be out when these animals are mating. And so you'll hear this handsome specimen right here calling. And then there's also some green frogs calling in the back. So again, because we are studying physiology, we need measurements of what the organs are doing with the blood, how the blood is responding. And so what we usually do is we collect the animals and then we keep them in the laboratory under conditions that, again, mimic or basically recreating what they would experience in their natural environment. So when we looked at the genes, again, in the liver of frogs, of tree frogs, when they undergo freezing, we found that genes that were associated with cryoprotectant breakdown were less active. We say downregulated. It basically means that you're making less protein from that particular gene. And so what this really means is that anything that would basically take away cryoprotectant from these animals, so anything that would cause the breakdown of the cryoprotectant, either for energy purposes or other purposes, is going to be minimized. So there seems to be at the gene level this tendency to keep the cryoprotectant around when the organisms are frozen, which would be beneficial to them, given that they need that cryoprotectant, again, to minimize ice formation, stabilize enzymes, etc. We also found that a lot of genes related to the stress response were activated, so their genes were basically upregulated. That's the term. So things related to heat shock protein response. Heat shock proteins are proteins that protect other proteins from damage. And then we also found genes associated with DNA repair. So cold and other stresses associated with freezing, such as lack of oxygen, and then too much oxygen when the frog thaws, can cause DNA damage. And so it was interesting to find that genes associated with a DNA repair specifically were turned on when the organism was experiencing low temperatures. But then surprisingly, genes associated with oxidative stress and anoxia response. So anoxia, basically lack of oxygen, and then oxidative stress has to do with the fact that these animals undergo periods with no oxygen, and then when they thaw, you have a lot of oxygen coming back in, and that can be also stressful and cause different types of damage. And the genes in that particular category didn't really respond. So there seems to be some involvement and some regulation of not only cryoprotectant responses, but also other protective responses coming from a genetic component, but that isn't the whole answer. And so then recently, what I've been working on is trying to see what exactly triggers cryoprotective responses in tree frogs. So we know that freezing triggers cryoprotectants. These frogs are peculiar because they don't always respond the same way to a freezing episode. So they turn on only certain cryoprotectants in certain freezing episodes. And I'm interested in understanding exactly the role of the various components of the freezing stresses in turning on these cryoprotective responses. So I'm looking specifically at dehydration, because again, freezing is kind of like a cold dehydration. So the animal is at sub-zero temperatures and water is being removed and turned into ice. So to do this, we again collect frogs from the fields, my students and I, and then we bring them to the laboratory and subject them to temperatures and photo periods that would be similar to what they would experience in their natural environment as the seasons progress. So we put them through a summer and then a fall and then a winter. But then we also wanted to see how water availability affected the cryoprotective response. So closer to the end of the fall, we started triggering a dehydration, and we triggered a dehydration basically by limiting access to water to these animals to see again if we could kind of tap into their ability to survive a restricted water environment. And then we basically compare the animals that were dehydrated with the animals that were not, to see again what kind of physiological response we are witnessing. And so we did this experiment twice. In the first year, we did a mild dehydration. And so the animals lost about 10% of their body water. And you can see that right here. So here we have water concentration in the tissues. So basically the proportion of water in comparison to the mass of the tissue. We have in blue the winter group, which is the control group. In white, we have the dehydrated animals. And we have here whole body water and liver water. So the goal was to create a dehydration that would be similar to the dehydration that these animals experience during freezing. So, because of the ice formation, again, 2/3 of the body water can be absent as ice. But as you can see, the mild dehydration was very mild because 10% of the body water is gone, but that wasn't even detected at the level of the liver. And so then when we looked at cryoprotectants produced in these animals, we see that there really wasn't any. So glycerol in the winter group, in the control group, was similar to the dehydrated group. Glucose still not different, and urea still not different. So mild dehydration really did nothing to these animals. And it turns out that tree frogs are very tolerant to dehydration. We used a dehydration protocol that had been used in wood frogs. And so that was not capable of inducing a dehydration response in these animals. So then we did the experiment the following year with a more severe dehydration. And we were successful in creating a dehydration that more closely related what would be experienced during freezing, not as extreme as what these frogs experienced during freezing, but still pretty intense. And so we found about, like a 30% loss in water at the whole body level. And then at the liver lost about 20% of water. And again, the animals are alive until the end of this process, so they survived fine, they were responsive. And then when we went and looked at cryoprotectants, what did we find? Well, surprisingly, we didn't find any response from glucose, but glycerol and urea, we did find an increase. So D is for dehydration and W is for winter. And so what you can see is dehydrated frogs produced massive amounts of glucose and also large amounts of urea. The urea was not surprising. This is a typical dehydration response in amphibians, the accumulation of urea. But the glycerol was quite surprising, as long with the glucose as well. So mild dehydration did not induce protective responses, but the severe dehydration did. And it seems like tree frogs seem to favor one cryoprotectant, glycerol, relative to glucose, which is peculiar and somewhat unexpected. So we are now trying to determine what exactly in terms of metabolism is directing these frogs to use one cryoprotectant versus the other. So we're looking at things such as enzymes and other proteins in the liver that could be related to this specific option, so to speak. And other things that we will be working on in the future include, so you remember how I mentioned how it's fairly difficult to go out in the wild and see real time these animals undergoing freezing episodes, and instead we do these in the lab. We're trying to, do a compromised approach with this, in the sense that we're building an outdoor enclosure. And so the hope would be to have these animals hibernating in a semi-natural environment, and then seeing if these responses that we see in the lab, such as, reliance more on glycerol and things like that, are happening when they're exposed to all the different environmental factors that they would if they were in their natural environment. So why should you care about freeze-tolerant frogs? Why am I doing this? So there's a couple of different implications for this type of research, and a couple of different reasons why this research matters, and why it will or may affect you. Maybe not today, not tomorrow, but maybe several years down the line. Fundamentally speaking, I do believe that there's a value in understanding how nature and how organisms work, and how they have evolved to deal with the environments in which they're in. So knowledge for the sake of knowledge. But from a practical standpoint, understanding how these organisms survive in these environments will also allow us, for example, to predict how changes that we are witnessing currently in our climate, and that we will continue to witness, will affect the survival of these organisms and their responses, given that they are so dependent on their environment for many of these responses. From a more medical point of view, this type of research has implications for cryopreservation. So we are talking about frogs that can survive being completely frozen, when us humans, we can't even freeze a human heart. So we are right now severely limited in terms of using organs for transplant within a very short time frame of hours, because the tissue can only survive for so long. If we instead were able to use some of these physiological responses and translate those into how to freeze a heart, a liver, a kidney, etc., we could potentially in the future do organ banking, where we would have, frozen organs, that were donated, and then that could be used by people that needed them. And to demonstrate that this is not far-fetched, I have two final things to point out. So here on the bottom left, I have the summary of a meeting that happened in 2015 called the Grand Challenges of Organ Banking, Proceedings from the First Global Summit on Complex Tissue Cryopreservation. So this was a meeting with prominent scientists all over the world that worked either on cold cryopreservation, and also scientists that do the same things that I and my colleagues do, scientists that study organisms that are naturally cold tolerant, freeze tolerant, etc. And so there was a specific panel in this meeting whose job was to basically summarize all the adaptations that we see in the natural world from these types of organisms, and basically describe them and explain them to scientists that are working on how to freeze a heart, how to freeze a liver, so that you can see how basic science could potentially inform applied scientists, so the scientists that are actually trying to develop cryopreservation strategies. On the bottom right, this is a logo from the Defense Advanced Research Projects Agency. It's also known as DARPA. It's a branch of the military, and they do a lot of different types of research, not only from the weapons perspective, but also from the medical perspective applied to combat and soldiers. And so they created this project about 2 years ago, where their goal was to extend the time available for helping soldiers that were critically injured in the field. And so they called it extending the golden hour. And so literally in the description of this grant project, this grant call, it's kind of like a competition for scientists. You write a grant and then they choose the ones that are the best and then give them money to do the research. And so their call, what they were saying was, we wanted to look at these extreme organisms, the organisms that can do extraordinary things like the wood frog, and take what you know from them and translate it to human cells, to human organs, to develop a technique, a strategy, or an approach that would allow us to gain more time in the field to help soldiers that have been critically injured. And so again, this was 2-3 years ago. So we are working on this. People are working on this. And again, taking basic knowledge from these organisms and trying to apply it to improving, human health and also help to predict other challenges that we may have in the future. So I did none of this work alone. I have been very fortunate in having multiple students, collaborators, and mentors that have helped me throughout the way. These are just some of them. I've worked at various different institutions that have all been very generous with their time and funding. And so I have all of these colleagues and mentors and students to thank for their continuous support and for helping me with my work. And I will now take any questions you may have. Thank you very much for listening.
Great. And if anyone has any questions, please go ahead and place them in the chat box. And I do see one in there from Liz, and she asks, are the wood frogs conscious during the thaw?
So that is a very good question. So we know that there's a specific timeline for the recovery during thawing. So one of the first things that happens during thawing is you have melting around the heart, you have circulation coming back, then you have ventilation coming back, and so eventually you'll have nerve conduction